ready to listen about some corn. So yeah, I'm Natalia de Leon. I am part of the Plant and Agroecosystem Sciences Department. That is a new department. You didn't miss much. It's that you have been on campus. It's a new department. I was part of the Department of Agronomy, who joined with the Department of Horticulture and chose the longest department name on campus, Plant and Agroecosystem Sciences Department. I had to practice many times. It is an absolute honor for me to be here. I mentioned to Jake, I am a plant breeder, which is not the same as a plant geneticist, and you're going to find out. I will talk as a plant breeder. I hope what I tell you is of interest and relatable to a lot of things that you are doing. And so what I'm going to talk about today is talking a little bit about variation and historical variation, and thinking about it from the perspective of a plant breeder, which means I'm thinking about it from a very practical standpoint, how we can use that information and how we can think about variation for improving crops. Down there is my email. If anybody has any questions and would like to get in touch with me at the end of the presentation, also pretty long email. I have long department names, long emails. I added these two slides after I submitted my deck to Marta, so I apologize if this looks a little bit different, but I realized that because I think about these things all the time, and because they are the motivation for a lot of what I do need to be very explicit about that when I talk to different groups. So I want to put my thoughts in context, and this slide is not to say that I feel like I have any power in solving all the challenges that affect our world, but to recognize that they are real and that they are the motivation for a lot of the things that I'm going to talk about today. So there will be 9 to 10 billion people on this earth by 2050, and we will be working with less water, more variable climates, and soils that are likely to be more degraded. There are lots of studies that have done lots of assessments of all of these issues over the years. This is my favorite. It's becoming a little bit outdated, but the reason why I really like this particular report is because it was able to quantify, making a lot of assumptions, but actually quantify what is the impact of all of these things in the way we need to think about food production and how we go about maintaining our ability to live in this world. So based on this report, they have estimated that in order to be able to feed these 9 to 10 billion people in 2050, we are going to need to produce 54 more crop calories in 2050 than in 2010, which is when the data was collected for these reports. If we use the 2010 rate, which is again what this report used, we are going to have to add twice the amount of the country of India to land that is in production for agriculture by the year 2050. And there is a very significant effort that needs to happen in order to be able to maintain greenhouse gas emission mitigation to the level of that below 2 degrees above pre-industrial temperatures, which is a goal that is already kind of on the edge of making things pretty hard, but that is what we are trying to work. So I know, you guys read a lot, and so these are not things that are new to anybody, but sort of to put things in context, that these are sincerely a lot of the motivation of why I do what I do, why I think about the things I do. And the thing about it is that, all of these things are talking about the year 2050, right, as the year when things are going to it's not magically change, but, we will have all those people on planet Earth. And so when you think about the kinds of things, again, that we do, and you think about the domestication and evolution of the crops that we have today, it took about 10,000 years to transform. Natalia is really bad at drawing and cartooning, so you're going to see the same little corn plants just kind of moved around, made larger, smaller, because that's about all I can do. But what you see here on the left is Teosinte, the ancestor of corn. And what you have on your right, my left of the screen is a really nice productive corn plant with lots of ears, looking very green and healthy. It took about 10,000 years for us to be able to move from teosinte to productive corn plants that we grow today. And we have about 10,000 days to be able to maintain that productivity or increase that productivity, while taking into consideration that we can't just do it like we have done it in the last 10,000 years. We have to do it with less water, less input, considering changes in climate, and using plants not as a way to just, not just to reduce the impact of plants, but to think about how can we use them as a mechanism to mitigate some of these issues. So, the clock is ticking. And that's a very important part of why, thinking, as I said, the difference between maybe a plant breeder and the things that I do. So, the field of plant breeding, it's very broad in the sense that we think about genetics, but again, we think about productivity, and we have to think about the climate. We have to think about technologies. It incorporates a very large number of fields in, in the decision making of what we do. But it's funny enough that it all kind of boils down to this equation, which very selfishly we call it the breeder's equation because we are just that cool. So the breeder's equation is the genetic gain equation that says that genetic gain on a timeline basis is a function of 4 different things. One is how intensively we select for certain traits. What is the variability that we have available to work from and with? How heritable is that particular trait? That means when I measure a phenotype, how representative that phenotype is of the genetic that controls it. And then how quickly I can circle through the life cycle of a species in order to be able to make more than one selection, for example, per year. So time is in the denominator, everything else is in the numerator of this equation, which means that the more intensely I select, the more variability I have, and the more heritable a trait is, the greater will be the gain that I'm going to make in these selections. So we do spend a lot of time and use a lot of technologies to try to make things, select more intensively, improve our ability to understand how genes control phenotypes. And then we do also a lot of work trying to expedite life cycles of these plants in order, again, to make more gain on a yearly basis. We do think about genetic variability, but my whole point in this presentation is to say to make the point that we are at a time where we maybe need to think about where this genetic variation is coming from and how we work with it in a slightly different way than we have traditionally done, especially, again, in the context of plant breeding. So I call these, and this is one of the papers that I share with the group, these breeding lottery. So again, breeding is based on the idea that we have mutation changes, our genetic constitution. Recombination uses that to create new forms, and then breeders exploit that to make improvements. But the ability to combine all of those events into what we, think about the ideal genotype is actually not as high as we typically think about. So very back of the envelope, simple calculation. If we think about the probability that if I create an F2 progeny out of, for example, 2 inbred lines, and I say, I want to identify the ideal genotype, the probability that I will identify that ideal genotype, which is the homozygote that contains those favorable alleles, it's about 1 in 4 if I'm thinking about one locus. If I think about 3 loci, that probability all of a sudden is 1 in 64. If you know where I'm going, right? I mean, if I say 5 loci, that is 1 in 1,000. If I look at a trait that is controlled by 15 loci, it's more likely that I will actually win the, what is this? The power jackpot. I know the probabilities are very, very low. Whatever that probability is higher than the probability that I will identify the ideal genotype if I'm trying to improve a trait that has 15 loci. And that's what breeders are trying to do. We are trying to combine the most favorable alleles across all these traits. And the vast majority of the traits that we work with that are economically important are very likely to be much more complex than 15 loci. And in this equation, I'm not even considering the fact that all these things interact in different ways. So for example, the genome of corn has about 32,000 genes. So just to kind of put things in perspective. So this is what creates what I call the conundrum of breeding. The conundrum of breeding is that plant breeding relies on genetic variation, but not all variation is created equal. If I want to create a sunflower, it's a lot easier to start with a combination that includes something that looks like a sunflower than if I start with all kinds of things that look very different from a sunflower. And this is me using ChatGPT and asking it to make some pretty pictures. They look very complicated, much more complicated than I wanted to. So thinking about variation and where that variation comes from. The variation that we have access to today is historical in nature. So here is me again, whether we are thinking about domestication, from the very small teosinte that improve into heirloom varieties that then became the corn of today's over 10,000 years of domestication, or if we think about the unimproved varieties of about 100 years ago compared to today, we see that Gaussian distribution is becoming narrower because that is the way that we get to those favorable alleles and we accumulate them in our improved varieties. So whether we are thinking about domestication or breeding, the variation is just a reduction. It's all historical. There is no addition of new things. And actually, there was some beautiful work that was published some years ago by Chuyan Cheng. He's a research scientist in my group. She did her PhD here with John Dobley. And I'm not going to go into the details of this work, but she showed by comparing two different QTL populations, one using teosinte, the other one using maize, and they had about, I think, 18 or 20 different domestication traits. And what that figure there shows on the orange bars is that the number of QTLs of the teosinte populations were larger than the maize populations, indicating that, what I'm drawing here with my Gaussian distributions is correct, that the amount of useful variation or variation has been reduced during the process of domestication. You might make the argument that there are lots of new mutations that are created every year. And of course, recombination is a machine that produces variation. I would argue that that is possible and that is real. If you think about, primarily if you think about every year in the United States, we plant 20 times more corn plants than stars in the Milky Way. So you would say, okay, look at all of that variation, look at all of that opportunity for recombination. The reality is that out of that very large number of corn plants, there is only a small, that was me again, me and my animations, yeah, a very, very small number of plants that are actually evaluated and actually effectively recombined and used that will contribute to the next generation of mutations and recombinations. So yes, there are lots of corn plants in the world. Not a lot of them contribute to that source of mutation and recombination to the next year. And I will not preach to the choir. When we think about mutations, if those mutations are neutral, yes, they will survive over a very large number of generations. And this is just, again, a typical picture that you will find in your population genetics books. So they will be floating around, but they will not contribute to the variation that we can exploit, the variation that will actually make changes into phenotypes that we are looking for. And even if they are not neutral in the environment that they show up, if they have a small effect, and I just, again, just Excel and me playing around with it, those small effect mutations, when they are at a low frequency, they will most likely be lost after a few generations, or if they are large enough, they will, either be lost a lot faster or they will be fixed a lot faster. So there's not a whole lot that is actively available for breeders to work with in populations. The other component of this is a figure that could look complicated, but I will try to walk you through it. So what we are seeing here is this is the description of 25 nested association mapping populations. So this is The reference genome, the first reference genome for corn was B73. This group led by Ed Buckler crossed that B73 by 25 different inbred lines, created 25 different recombinant inbred line populations of about size 200. So this population is a population of 5,000 recombinant inbred lines that have one parent in common and 25 other parents that represents the diversity of corn, to the best of our knowledge. And what you have in here depicted is the 10 chromosomes of maize are listed horizontally, and then you have the 25 populations. And the different colors just show the proportion of B73 alleles in these different 10 chromosomes across the 25 populations. You can see how the colors change pretty rapidly or drastically in some of those sections that are highlighted, either in bluish colors or reddish colors. What that shows is that when they did this study, they found that about half of the largest QTLs that were associated with yield in this study, this very large study that they did back in around 2010, were actually located in the centrometic regions. What that means is that those are regions with the lowest amount of recombination. And what they actually found is that when they compare residual heterozygosity in the centromeres compared to the rest of the chromosomes, there was a greater proportion of that residual heterozygosity in those centromeric regions. I could spend a whole hour just talking about that relates to the theory of the dominant theory of heterosis and how that has been proposed to explain a lot of the yield or the heterosis that we see in yields in hybrid crops such as corn. But the idea here is that just to show that this is another thing that we also need to think about, is that recombination doesn't happen uniformly across chromosomes. And it just so happens to be, we can have arguments about whether that's biology or just coincidence, that a lot of these important QTLs that control important things like yield are in repulsion phase linkage, and they happen to be in areas that there is very little or reduced recombination. Okay, so this makes things even a little bit more complicated. And this study is probably 14, 15 years old. I just added this other more recent piece of work that was published last year or 2 years ago. They did a telomere to telomere assemblage of the maize genome, and what they found is that 82 of the most important or very important genes that were identified in this work were actually located in the centromeres. And the reason, they show the importance of these genes by showing the homology of these genes with other, within the maize diversity collection and also in sorghum. So showing that these genes, they do tend to accumulate in these areas of low recombination. So what have I said so far? So variation is the substrate of breeding. But we think about these, and when we think about these, we need to realize that large positive mutations are rare. If a mutation is beneficial in an environment, it is more likely that it will be fixed, or it should be probably fixed by now. So it is somewhat improbable that we have new foundational variation that is currently available in breeding programs. And most of what we have is the consequence of historical variation that, it stays there, and that is with the predominant substrate. So what we have are probably just a collection of very small effect alleles that are linked and probably in repulsion phase linkage. That's sort of the foundation of what we have to work with. We are not fully depressed yet, but this is very likely the situation. Okay, you can also say, okay, there are other sources of variation. Corn is notorious for the presence of a very large dispensable genome. So there have been lots of studies looking at the pan genome. So there are about 32,000 core genes in corn. There's about 70 or more thousand dispensable genes. So these are possible source of variation, and they are. But I will tell you some of the work that we have done. This is, I'm summarizing a whole bunch of different papers and things that have been published over time and a little bit more, but again, work that we have done in our group looking at 80 different tissues representing the timeline of growth of maize from germination to senescence. We did everything the way it should be, replicated samples, you know, you name it, compare everything to the reference genome, B73, and, you know, so doing that, like this very comprehensive RNA-seq atlas of the development of corn. What we found, looking at this type of data, is that when we compare what you have in here, the blue represents the comparison to the B73 reference genome. The green represents the reference to the another inbred line, PH207, and the two comparisons are showing more or less the same idea. Is that when you compare the size of these genes, looking at genes that have presence-absence variation compared to the core set of genes, whether you're comparing it, you know, B73 as a reference or PH207, the genotypes are, the genes are slightly smaller and in general less complex in both, you know, in the than the genes in the let me try again. The presence-absence genes are smaller than compared to the core genes, whether we are looking at B73 or PH207. We also look at the expression of these genes, and in here, what we did is to compare. Here, we are comparing blue and red. The two on the left are comparing B73 specific, and we are looking at expression, FPKMs, on the y-axis. B73 specific and present in B73 and PH207, for example. And then the blue, the farthest blue represents presence-absence in the 32 lines. This is a collection of 32 lines that were evaluated, and then presence in the 32 lines. So again, comparing pan genomes versus core genomes. And in both cases, the expression of these presence-absence genes is lower than the core genes. So these genes are smaller and they are less expressed. And when these genes are expressed, what we found, and I'm going to just put the whole idea here, the graph on the left are the genes that are present in all 32 lines that were resequenced. The ones in the blue are PAVs among those 32 resequenced genes. And what I'm showing here on the x-axis is the number of tissues where they were expressed. So this story shows that things that are present that are present everywhere, they are expressed in a lot of tissues or all tissues. Things that are you know, expressed only in some of the lines, they will also be expressed in one or very few tissues. And this same type of work, I mean, again, this data is a little bit outdated. Just to show you, this is a paper from last year, beautiful work that Matt Huffer from Iowa State published last year. And my circles over there are a little bit small, but the one on the left is the core genes, and all the other ones are pan genes, and the darker the color shows the more tissue. So kind of confirming the same idea, that these genes are smaller, they don't express as widely, and when they get expressed, they express in a limited number of tissues. So some dispensable genes are useful, but many are not functional, and they are likely to be lost. And then, you know, the other thing that as breeders, everybody says is like, well, but you have all these tools that allow you to dig deep into the variation that you have available. You have abilities to not just base yourself on, you know, the variation that is available, but use these tools that help you cycle through generations of breeding much faster. And one of the most widely used technologies is what we describe as genomic selection. So genomic selection is the idea that you can take a population, you can phenotype and genotype that population, you can train models based on that population, making connections between the genotype and the phenotype, and then you only need to genotype other populations without the need for phenotyping, which allows you to crank through the breeding pipeline and be able to theoretically explore and increase the frequency of favorable alleles much faster. Of course, there are lots of factors that affect the efficiency of these methods. The bigger the reference data set, the more heritable the trait is, the number of loci that affect the trait. But one of the most important things that makes these tools be effective is that the closer the training population is to the testing population, so the closer genetically the population that you're genotyping and phenotyping it is to the other one that you're trying to improve only using genotypic information, the better off you will be. So this is data, like 4 different traits. These are not plants, these are animal traits, and I'm using this paper just because the graph is, in my opinion, the clearest. They use 3 different models to show, but the idea here, just to show what I said, is that the closer the 2 sets are from each other, so the relationship to the right shows increased relationship, the more related those training and testing sets are, the higher is your prediction ability. So what this is saying is that even the technologies that breeders are using consistently and deploying consistently tend to make the variability that we have access to even narrower. We are taking that variation and decreasing it or driving to the goal even at a faster pace. So it's just an accelerated or improved version of the lottery. Let's run faster to a particular set without really bringing any other variation from anywhere else. So the title of this slide is Infinitesimal Model is a Consequence, because when you think about all the things that I just told you, it is not coincidence that a lot of the economically important traits, or the traits that have been selected for a very long time, seem to be controlled by a very large number of a small size genes. And so the argument that I'm trying to make here is that it's not biology, it's more likely just the way we do things. So genetic variation is required for breeding success. Most existing genetic variation is not beneficial in any context. Good or bad variants, they will be filtered by breeding and or evolution. And then the recent variation that is likely to be available is neutral or negative. And of course, again, I started this presentation by saying I am a breeder, so I know, the context of that, whether it's neutral or negative, it's very important, right? The environment changes the value of an allele in a second. So minimal new variation is arising in breeding populations. So this is a sentence, Sean Kepler, who's a colleague of mine in the department, and I shared nurseries and fields. We spent a lot of time in the summer pollinating corn. And one of these days we were there 100 degrees, and Sean turns around and is like, you know what? I was just thinking, this infinitesimal model of quantitative variation is not the result of biological constraint. And I'm like, oh, wait a minute. This is not the time nor the place to be telling me something like this. So I wrote it down, and then I thought about it for a while, and that was the inspiration for a lot of what I just told you. So lots of crazy things happen in cornfields. And the thing is like, okay, and all of these will not be that relevant if, as a breeder, I wouldn't have the urgency to know that I have 10,000 days to make the progress that is needed, considering all the challenges that we have ahead. So we need new poles of variation that will maximize breeding success, and think about things like creating larger effect alleles, creating desirable intermediate effects, considering how we can diversify gene function. You guys in the audience are the experts. I mean, can we think about cross-species transfer of gene function? Can we create targeted positive variation that could be a new substrate for selection? And of course, it will include altering recombination frequencies, unlocking variation, you name it. So this is not earth-shattering. I mean, this is obvious, right? The thing is that until very recently, we didn't have the ability to really do these in things like corn or soybeans or other economically important traits. We have the ability for a long time now to create transgenic plants, but that type of modification is quite random, and so you are just adding a little bit. In many cases, you can use those technologies, excuse me, to make relatively simple changes, but you can consider those technologies to make the kinds of changes that will impact traits like the ones that I'm normally thinking about. So I'll tell you a couple of examples of things that we have started doing, and then I want to sort of transition into thinking about what are the constraints and what are the things that from a practical standpoint we are thinking about in this context. So this is, so I'm going to talk about a collaboration that we had with Jonathan Lynch. He's at Penn State University. He has spent his whole career thinking about how we can change roots to increase the ability of roots to acquire input such as nitrogen and water. So he has these motto, which is create roots that are steep, cheap, and deep. So you know, the farther, you know, the more the roots grow into the soil, in occupying the least amount of space possible, and then in the cheapest way possible, thinking about parenchymas and you know, ways to minimize the metabolic cost of these roots, the more likely it is that those plants are going to be able to acquire those inputs in an efficient way. So he spent his whole career thinking about how to do that. And a very important part of that is creating roots that have steep growth angles. We have done numerous experiments and literally across decades in the United States, Africa, South America, looking at different sources of variation. Very long story, but this is a GWAS plot of probably one of the most successful GWAS analyses that we've done in roots. And I know you're going to say, this looks awful, right? There's one dot above the threshold. And that is just to say the complexity of these traits is very, very hard. These are very noisy. The phenotypes are really difficult to evaluate. But yes, we identified through forward genetics that one site in chromosome 1 that had a significant SNP with an effect of 2.6 degrees, literally 2.6 degrees change in the angle of those roots. Because of the ability to edit plants, that gene, that SNP was identified to be in this CIPK15 gene. Through reverse studies, we were able to then modify that gene to create a root angle of 10 degrees, more or less. And the most important thing is that under suboptimal conditions, that gene allowed an increase of 18% on shoot biomass and 29% on shoot nitrogen concentration. A very significant increase in the goals, the practical goals of what we were looking for. And again, it's starting from a gene that had a very small effect, and then the ability to bring that gene into the context where it made an important impact on an important, economically important trait. Another example, so that is taking a gene and causing a larger effect. This is another quick example, editing the brown midrib gene. So being in the dairyland silage, or so let me back up a little bit. So dairy cows in the state of Wisconsin, where it's very cold in the winter, their diet is based on eating corn. A lot of that corn, there is some corn grain that is fed to the cows, but there is also a fair amount of corn silage. Silage is whole plants of corn that are chopped by the farmers, fermented. If you drive around the countryside, you might see those tall blue towers in the fields, or the long plastic bags that are sitting on the side of the field. Those are bags that contain silage. It's pickled corn that farmers feed to cows during the winter, and it's very, very important to keep those cows producing the enormous amount of milk that they produce year round. So very long story short, digestibility, or how easily those cows can eat that biomass, is really important. So we spend a lot of time making corn plants more digestible. Making them more digestible means that we are putzing around with the cell wall of those corn plants, making them less lignified, and in some senses weaker, right? I mean, you take the lignin out of a plant and the plant flops. So we are doing everything that probably instinctively will say, no, don't do that. I mean, you want plants to stand up, you don't want them to fall over. In order to have more digestible silage, you need to have that higher digestibility. You have to have modified forms of lignin. So this particular gene, brown midrib, produces that increased digestibility. As you might imagine, it improves digestibility, but at the same time makes these plants also weaker, more prone to lodging and so forth. So what we want to try to find are versions of this gene that produce increased digestibility, but doesn't allow the plants to fall over. That's the short story of this. So we actually were able to identify, we think, some of those. I'm going to make a very long story short, but through editing, changing these alleles, creating some mutations that created reading frame shifts versus just, a small number of SNP mutations, we were able to create contrasting types of alleles. So what we call weak allele and strong alleles. And we were able to compare. I should have started by saying that this gene is called brown midrib because it creates a brown midrib. So the midrib of the wild type is green up at the top. The midrib of the strong allele looks pretty brown here at the bottom. And then you have that weak allele that is intermediate, even, to the eye. So what we did is to take that weak allele, strong allele, and the wild type and compare it. And what we are trying to do, I'm going to have to walk a little bit because I don't, this is a picture that if I don't walk you through it. So what you see over horizontally is a corn stalk, the main part of the corn plant. And what you see on the top is a machine. This engineer in front of us has a machine that actually pushes the stalk down right at that breaking point, and is able to measure how much that plant is able to tolerate the breakage. And so we want plants that stay steady because we want those plants to survive the entire season standing up. So this is the example of a Imber line that we have where we backcross brown midrib, just, allele, the wild type and the brown midrib. And what you have in the y-axis here is the stiffness. What happens, when that thing is putting pressure on the stalk. The wild type is much stronger than the brown midrib, and that's what we see across the board in this type of comparison. What I'm showing you here are two wild type lines, the LH244. These are a little bit improver lines, so they are by nature stiffers. And what you see here is the comparison of the strength of the brown midrib, strong allele, and weak allele. These two alleles have the same digestibility value, but they are much different in their ability to sustain that pressure. So for us, this is great. That's what we want, right? We want the modifications that allows us to get what we want without losing what we don't want, right? Or the other way around. You know what I mean, okay? So this is just one example of making modifications that are guided by us so we can get to the variability that is of interest. So you might ask the question, why we are not doing this all the time? I sort of alluded to it already. The technologies for complex species or individuals like corn have become available fairly recently. And the most important thing is that for the technologies that allows us to direct modifications in these corn plants, a lot of those tools only worked in very restricted types of genotypes, which are usually pretty poor, not things that you're going to be usually using in a breeding context. So now we are able to edit virtually any genotype that we want. And that, in the context of a plant breeder, is a huge difference because I can improve, with all due respect, I love Jerry Carmichael and W22. But as a breeder, I can improve W22 with my eyes closed, right? Because I don't know if you have ever seen, I'm looking at the corn breeders. W22 is a very little, weak, very hard to work, doesn't like to be pollinated kind of corn plant. That's not what we want as corn breeders. We want other types much stronger, much better. So now we are able to edit any plants that we want. So up until recently, there were lots and lots of workshops in national NSF, DOE organized workshops because everybody was very focused on bringing the technology. The limitation was we don't have the technologies that we need to be able to make these modifications that would allow us to make progress. We are past that. Those technologies now exist. Actually, Wisconsin is home to one of the most important public centers that do tissue culture transformation editing of about 22 different economically important species just on the west side of town. It used to be a facility owned by Monsanto Bayer that is now part of the University of Wisconsin that provides services not only to our campus, but to the community at large. The largest center in the United States. And so the perceived idea that the limitation was our ability to transform plants has now become that the real limitation is not the plant, not the transformation, but how do we know what we want to change? And then once we change it, how do we know we made the right changes? And both things are important. I am running out of time, so I will be very fast and probably jump through a few things. I wanted to kind of show you an example of, I told you that a big focus of what, as a plant breeder in today's world, I worry about is how do we improve these plants, not just to improve productivity, but to improve productivity in the context of, the sustainable agriculture needs that we have. And nitrogen is a very important part of that. Nitrogen has been very, very important to improve productivity, and we wouldn't be here if it wasn't for that. But, there are issues with water pollution, there are issues with soil contaminations, and you name it. So I am going to go through these two slides very fast because just to go to these, describe very briefly this project that I am involved with, again, led by Ed Buckler, called the Main Circular Economy that Reimagines Corn Agriculture. Talk about long, today I'm the lady of the long names. That is the name of the project, also known as CIRCA. And what CIRCA is trying to do is to harbor alleles that are necessary to improve nutrient recycling and reduce the nitrogen contamination issue that is highly associated with annual crops, especially corn. And it is a bit of a longer story, but I will just give you the highlights. So a lot of the focus of this work is reduce the amount of nitrogen that we find in corn kernels. Currently about 8%. Corn is not an important source of nitrogen or protein for animals, so there is no need to have all that protein in the grain. Second is to improve corn. So we have earlier germination, even in colder places like Wisconsin. So we have biomass covering soils during times of the year where once the snow melts and there is no green coverage of soils. In Wisconsin, for example, that's the time of the year where a lot of those gases coming out of the soil move from the soil to the environment, creating great fluxes of greenhouse gas to the environment. So if we are able, sorry about that, if we are able to plant corn, for example, earlier, that green biomass will prevent those gases from moving from the soil to the environment. And then lots of works on trying to sort of transfer the nitrogen recycling mechanisms found in perennial species to an annual crop like corn. So there is a lot to unpack in there, but this is the goal of this project. And of course, all of these are ideas that we are, theoretically thinking about, but everything that we do is like, okay, I'm going to, for example, decrease the amount of nitrogen in the kernel of a corn plant. If I plant that corn plant in a very nitrogen-rich soil versus a very nitrogen-poor soil, the response and the phenotype would be very different. And different genotypes will react to that different environment in different ways. So when we are thinking about all of these changes, and I'm gonna, sorry, never do that when you're giving a seminar, but here I am doing it. This is the part that, that's why this arrow becomes red, because we can't only think about what are the changes that we want to make. We have to think about what are the changes that we want to make in the context of the environments that we want to target. And there are lots of them, right? And so how do we think about that genotype by environment interaction? And how do we know when things are important where? So my advisor, my PhD advisor, used to say, you get what you select for, and that's the only thing you need to know as a plant breeder. Of course, he was joking, but what he meant by that is that if you select for something that is constitutively expressed versus something that will respond to, the cues from the environment, it's a very different beast. So if I'm thinking about flowering time, a root angle, the leaf of or shape of the leaf shape or angle, I can do that anywhere. I can select for that pretty much anywhere. If I'm looking about stomatal response, ovule abortion, nutrient remobilization, the context that I select for that will be incredibly impactful. So that's where I have to think about these genes in the context of where I want to put them. So I made that arrow at the end thicker because as we think about all of these important challenges that we need to address, knowing what are the genes that are important and the ability to modify them is one thing. The real bottleneck now becomes, how do I intelligently decide where those genes will be the most useful? And we have a limited number of opportunities to test all these things. One of my last few slides, I sort of, there was a group of us that got together, and this is, I mean, it's going to sound as a criticism, but it's not a criticism. It's just an alerting people that are thinking about some of these things. So this is actually a paper published last year in, I think, Nature's Communications or so. So this, what you have in here is the number of publications between 1990 and 2022 that investigated the impact of modifying a gene or a few genes and the impact of them in yield. And it's basically, the whole point of the paper is just to say that improving something that is not very good is very easy. And people can make get a lot of miles out of just taking something out of context and not really describing what is the real impact of that modification. So what that paper is basically saying, again, it was hard to write it not to sound like a criticism to the people that were reviewing the paper, that we needed to publish. This was very weird. They were offended, but they had to let us publish it. Is that when we are thinking about how to modify genes and actually talk about the impact that those genes might have in practical context, those studies should use a standard definitions of, for example, yield. Those trials should be replicated across geographies, locations, years to represent the true environmental impact. The varieties, the densities, the conditions in which they are tested should represent the real world. There should be proper controls. And then the researchers should prioritize genes that plant breeding might have missed, not go back to things that we have tested a million times. And that was one of the hardest ones to describe. But, this is all very important because now, again, we have the ability to make all these changes and we have the ability to do all these things, but we can't just go crazy and not thinking about what is the important context that happens. And the next couple of slides is, just showing an attempt. This is a very large multi-investigator, multi-state effort that we are coordinating here out of Wisconsin. Has been going on for about 10 years, where we actually test the same collections of diverse materials across all of these places with the idea to create standards, to create rules, and how, do it across years and across environments. And again, too much to talk about. I'm gonna just kind of finish up here by saying, this last part of what I was trying to say is that, we know how to make these modifications. A lot of the constraint now have become, how do we develop informed or well-informed hypotheses of what are the gene candidates that will make the biggest impact, and how we can create a context where those modifications can be tested in the proper conditions to address the needs of the crops of the future. And I have to just finish by saying that, again, all of these things towards the end especially sound like as a criticism. What you're seeing in here is the improve in yield average on the United States from 1920s to today's. And I always put this graph up because to me it shows the incredible impact of genetics, the application of genetic tools to breeding and how amazing it has been in terms of improving crop plants to, for the good of all of us. And then like, the challenges ahead. So with that, I will want to thank you for the opportunity, funding agencies, and then the group last summer. That's Jerry Carmichael right there. He is pollinating corn with us, and it's just such a gift. So thank you for the opportunity. And if there is time, I will love to answer any questions you might have. Uh-huh. I mean, that's a great question. I mean, it complicates everything, right? So for example, we are, a lot of these projects that I'm talking about that we are trying to do and creating all these modifications, we are limited on our ability to, it's a lot easier to knock down a gene. In order to knock down a gene and have a favorable phenotype, is not trivial. So that's where you need an intelligent design of hypotheses to create that situation. And when you're dealing with a hybrid crop, like for example, corn, you have to do that in two different backgrounds sometimes, depending again on sort of, how that gene is expressed. Because if you don't do that, none of that has meaning. And that's part of that last graph where a lot of that, not the last, I mean, what the paper that we published that shows all of those papers that have, it's not falsely described, but it is at a context description of improvements in yield. A lot of those are done based on inbred performance. Inbred performance is absolutely not representative at all of what, hybrids look like. And so that is not useful. That is not advancing real, solving real problems. And it is, I mean, the scientific aspect is very interesting. The argument in there is that you can't claim that that is, going to feed the 10 billion people living on Earth. Yeah. 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 But these things are the function state, but that's why yeah. Oh, yeah. I mean, they're all of the above. Yeah, by all means. And so, I mean, so historically, so corn breeding is done. So again, millions of details, but corn breeders have these pre-defined framework that they think about. So there are the concept of heterotic groups. So back, 150 years ago, somebody had to decide how to test these. They discovered that, when you created a hybrid, you have higher productivity than in the inbreds. So when you're, as a breeder, if you try to think about all the possible combinations, if you have 100 inbreds and you say, I want to create all possible hybrids, it's a very, very large number. Like it's impossible to test all of those things. So somebody had to make the decision to say, okay, I'm going to put 50 on this bucket, 50 on this bucket, and I'm going to just make crosses between. And that's how heterotic groups were created. It's not a magic thing. I mean, it's just logic, right? Somebody had to make a decision of what to cross by what, and then you start creating complementarity between these, regions in the genome that when they come together create this kick over inbred performance. And so that has maintained some of those blocks fairly constant. So it's not a bad thing. It's not a bad thing. It's just that if you take those blocks out of that context, then all of a sudden things start to fall apart. That's why, again, in the commercial world of corn breeding, people are very nervous about trying anything different. We don't want to bring too much variation or things from crazy places because you break that relationship. And and we write books and books and books about heterosis, and all of those theories are probably part of what we see. But a big part of it is that historically we have created these blocks of genomic regions that that they keep finding each other in these hybrids time and time and time again. Yeah. Yeah. Yes. Ah, yes. 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 So the typical case, the later flowering an individual is, the higher yielding it is. And it's, again, not magic. What happens is you have the plants will be greener longer. Greener longer means higher yield potential for as long as it doesn't freeze at the end of the season. That would be realized yield. The problem is that you can only go in that direction so far until your plants freeze in the field every year, and then you go from like having the potential of having wonderful yield to having nothing, right? And so pleiotropic could be from a genetic standpoint, and it could be just from a physiological. I mean, it could be not one gene. I guess what I'm saying, pleiotropic, not from one gene, but the correlated physiological relationships that exist on all these things. And that's sort of a very obvious one. I mean, in flowering time, physiological maturity is correlated with a lot of traits: yield, digestibility, standability, disease resistance. And it's not it's not something that you have to go genetically, that necessarily is like a one gene that I have to, figure out how to modify. Is yes, if you select for later plants, you will get all of those benefits. But we live in Wisconsin, so for good or bad. So yes. Yeah. Thank you.